Welcome, my name is Gregor, and I will be presenting a poster about splicekit, a comprehensive toolkit for splicing analysis from short-read RNA-seq. This work is coming from a small team, the spliceosome focus team at Roche in Basel. It's a co-first authorship with me and Arne Velik, and Arne will also have a talk about splicekit. I'm super invited to attend. If you're interested in more details, you can check out the preprint on bioRxiv, and also splicekit as a completely open source software. We have a GitHub page, and you're super welcome to check it out, download it, and try it. So the poster looks like this, and in this presentation, I will have only 2 slides. The first slide will be talking about the input to splicekit and really the basic analysis, the base of the data. And the second slide, I will be talking about 2 modules from splicekit, scan RBP and Djon. I would say the core of the analysis. So what is the input of splicekit? It's really short reads aligning to the reference genome. And for example, if you have a gene here with 3 exons, you could classify these reads into 3 categories, for example. The junction reads, so this would be reads here in blue, spanning exon-exon junctions. Reads that are specific to an exon, if, for example, for exon 3 here in green, and then all the reads together. For splicing analysis, we take the junction reads and use HR by providing it also the gene context. So we really want to see differentially used junctions and not so much differentially expressed genes. We do a similar thing on the exon level, so all the reads of exons, and then we also take all the reads and just do the classic differential gene expression analysis. The power of splicekit, I would say, is also that's very integrative, and for example, it provides a genome browser, and here all the data sets I will be mentioning are publicly available. For differential splicing, for example, you could visualize immediately the coverage of the reads for different samples. Here we have 3 control samples and 3 TDP43 knockdown samples. TDP43 is a protein involved in neurodegenerative disease. And for example, here splicekit immediately identifies a cryptic exon that is not expressed in the controls. Then what we do is also we try to understand if a treatment or a compound is influencing more splicing or differential gene expression. And for that, we construct judge plots, so junction differential gene expression plots. Here, a data set from Branaplam. Branaplam is a small molecule that's really a splicing modifier. And you see here on the horizontal axis, there is gene log-fold change, and on the vertical axis, there is junction log-fold change. And if you have a compressed plot, that would more signify a splicing modifier. And if you would have a wider plot, that would lead you to think that you have a treatment or compound that is more changing the expression of genes and not so much changing splicing of the molecules. And another thing you could do if you have several treatments, you could do clustering at the junction, exon, and gene level in terms of log-fold change. And again, you could see which treatments are more similar in these characteristics. And now really to the juice, I would say, of splicekit. One level is scan RBP. And here what we did, we took the positional weight matrices of different RNA binding proteins from a database called mCross database. And this allows us to take a sequence and then take a protein or all the proteins and construct a signal that's called binding prediction. It's a log odds signal and on a set of sequences. And then we take splicekit and we take the junctions, we identify the differentially used junctions, and we take the donor sites of the junctions and the 80 nucleotide upstream and downstream of the donor sites, and we classify these sequences into enhanced, repressed, and controls based on FDR and log-fold change. And then we take these three sets of sequences and we plot the binding prediction for TDP43 again, in this case. And here in the middle, we have the donor splice site, and again in blue repression and in red enhancement. And you see, which is also what is also very well known for TDP43, that compared to controls here in yellow lines, binding of TDP43 prevents donor splice site usage. And Djon, so donor junction anchor analysis, here again, we take the significantly changed junctions, but then we add another layer and we construct anchors. Anchors are 50 nucleotide regions at the donor site. And then we do counting of reads again in the anchors and do the analysis of the anchors. And together, the junctions and the anchors, we can really identify exons that are included or excluded in a cassette type of way. So a stricter, more powerful analysis, because junctions really guide us to where the action is happening, and the anchors help us to recuperate a little bit of power there, because the reads that span exon-exon junctions are usually few. And here again, we classify into included, skipped, and controls. And on a data set of a compound branaplam that I mentioned before, which is a splicing modifier, and is also very well characterized in terms of motifs that guide this splicing. And actually, if we look at the included sequences and compare them with controls using Dream, which is also an open source software for motif discovery, we find exactly the motifs that are characterized for branaplam. So this was it about splicekit. Super welcome to visit the poster. I think it's going to be 1287, if I'm correct. And welcome to reach out so we can chit chat live. Also welcome again to see the preprint. Welcome to check the GitHub page to download the software and try it out. And also please, if you like, also attend the talk on splicekit that Arne will be giving. Thank you for your attention.